Okay, so this is going to be part one of the muscular system section or chapter. This is going to be primarily about generally the types of muscle tissue and also muscle contraction. That's where we'll kind of work our way up to. Okay, so first thing again is the basic types of muscle tissue. We have three types of muscle tissue, and we've mentioned these before. This chapter will primarily focus on the function of skeletal muscle and how it contracts. And of course, for cardiovascular system, you'll talk more about cardiac muscle and smooth muscle. We talked about in several chapters as well. But for each of these, what I encourage you all to do is to find something that is unique to each of them so that you can identify one from the other. Number one, as far as the skeletal muscle, is going to be the only type of muscle tissue that is going to be voluntary, which means that we can consciously control it. Of course, skeletal muscle is called skeletal muscle because it's attached to the bone, attached to the skeleton. It's also striated, and we'll mention what that means later. However, striations will not be something that is unique to skeletal muscles because cardiac muscle is also striated. As far as cardiac muscle, it is involuntary, as is smooth muscle. But the thing that kind of makes it unique is that it has these branching type of cells. They kind of branch off. They also have these structures called intercalated discs that help to join one cell with another. And these are, of course, this cardiac muscle is going to be found in the heart. We have smooth muscle. Smooth muscle is going to be found in some of our internal organs like the intestines, the bladder, the the uterus. All of those will be considered smooth muscle. Also, the blood vessels as well. So our capillaries and veins will all be made up of smooth muscle. Okay, as far as skeletal muscle, again, we've mentioned these few already. And within skeletal muscle, we're going to have a little bit of nervous tissue because it's 100% connected to it. It also has a blood supply. And we're going to talk about the connective tissues that help to surround muscle tissue to kind of hold everything together and to form these levels of organization when it comes to going from a large muscle to a muscle cell. And that's where we'll start first. So around muscles, I'm sure you all have heard of tendons before. A tendon is going to be a structure that joins a muscle to a bone. Fascia or fascia is going to be this layer of connective tissue that surrounds a muscle. And you maybe have seen this before if you've ever maybe cleaned chicken, raw chicken, and you have this kind of white film over the top of it. That's going to be or clear white film over the top of it. That's going to be the fascia. I'm not talking about the skin of the chicken, but after you've skinned the chicken, sometimes you have this kind of thin layer that's going to be the fascia. And so over all of our muscles, we have a bit of fascia there. We also have the structure called an aponeurosis. And aponeurosis will be responsible for, it's more of a sheet-like connective tissue that connects one muscle to the next. For instance, the frontalis muscle is connected to the occipitalis muscle in the back of the head via an aponeurosis along the top of the head. We also have aponeurosis that covers the rectus abdominis. So, very important part here. So, we have three different muscle coverings going from largest to smallest as far as the things that they cover. The very first is the epimysium. The way that I kind of remember that is epi, of course, like epidermis, means outer. And so it's going to be the whole muscle, the very outside. And this is going to be found under the fascia. So if you look at this picture here, this kind of white tissue is going to be the fascia, this part that's over this here, that's going to be the fascia. But this outer layer right here is going to be the epimysium. So this kind of pinkish layer is the epimysium. And that's going to be around the entire muscle. So this was the rectus femoris over the femur bone. That's what the epimysium is. And if you notice, within the epimysium, there are several bundles. And so this picture is showing one of those bundles being taken out. And this bundle is called, this entire bundle is called fascicle. And it is surrounded by tissue called perimysium. And that's the tissue that's on the outside layer of this. That's the perimysium. Okay? And again, if you notice, even with the fascicle, within the fascicle, there's a bundle here, a bundle here, a bundle here, a bundle here. They're made up of bundles, and this picture is also showing a bundle with that being taken out. This bundle here is called a muscle fiber. This entire thing here is called a muscle fiber. And the muscle fiber is also surrounded by something called the endomysium. Endo means inside, so this is surrounded by the endomysium. And to take it a little bit farther, because we're going to go into these structures, again, if you look inside of the muscle fiber, aka the muscle cell, so this is the muscle cell, which, you know, looks a lot different from other cells that we may know. There are bundles of things here. All right. These are called myofibrils. We'll talk about these shortly. And the myofibrils, if you look here, are made up of even smaller structures, and these are called filaments or myofilaments. And we'll talk about these myofilaments a lot, because these are what's going to be responsible for muscle contraction, and the way that they interact with each other is what allows for muscle contraction. So, again, a muscle fiber. Muscle fiber is also the muscle cell, and that's this structure right here. So we're going to look at the muscle cell a little bit closer. Now, muscle cells are going to be very unique in that they have the same structures as a regular cell, but they are very specialized because they have to be ready for a muscle contraction. And so one thing we know about skeletal muscle specifically is that it is multinucleated. That's going to be something that is unique to muscle cells. It also has a structure called the sarcolemma. Sarcolemma is just like the cell membrane of any other cell, but because we're talking about a muscle cell, we have this sarco part. Sarco means flesh or muscle, and so that just means that we have the cell membrane of a muscle. Okay? And then we also have our sarcoplasm. Plasm may sound familiar because this is just like the cytoplasm. Again, kind of the same function, but it's specialized because it's inside of a muscle cell. And as with the last slide, we're going to go back. This muscle cell is made up of myofibrils, and those myofibrils are made up of myofilaments. And so again, muscle cell or muscle fiber made up of myofibrils, and those fibrils are made up of myofilaments. Very important thing to remember here, we have two filaments. We have actin, which is the thin filament, and we have myosin, which is the thick filament. We'll talk about these two a lot because those two are what are responsible for muscle contraction. Another thing that's not interesting, but I guess it is interesting, but is special about skeletal muscle that you don't find in smooth muscle is that they have striations. If you look at a microscopic view of muscle, skeletal muscle, you will see these kind of stripes or bands in the muscle, and that is due to the arrangement of the actin and myosin. So sarcomeres, or those striations, are due to the way that actin and myosin arranged. Remember, actin and myosin are those filaments. So the way that actin and myosin, those are the little kind of pink and purple lines here. The way that they are arranged are going to cause these kind of band-like structures. And we're going to look at a sarcomere very closely in a few slides, maybe even next slide. Okay. In addition, we have a structure called sarcoplasmic reticulum, also a very, very important structure. The sarcoplasmic reticulum is going to be responsible for the storage of calcium. And calcium is going to be a huge player when it comes to muscle contraction, because if we did not have calcium, then we would not have muscle contraction at all. And then our transverse tubules are just extensions of the sarcolemma. This is going to be what allows for nerve impulses to travel down the muscle cell. Also, triad, again, just another term of the tubule and parts of the SR. And the sarcoplasmic reticulum, just like the ER of a regular cell, but because we're talking about a muscle cell, it's very specialized. Again, very important when it comes to storing calcium. So, again, looking a little bit closer into these muscle fibers and the arrangement of actin and myosin. Remember, the myofibrils are made up of myofilaments, and this striation pattern is specifically because of the arrangement of those myofilaments, specifically actin and myosin. So the way they kind of are arranged here causes those stripes and striations. And so within a sarcomere, that structure that we call a sarcomere, again, that is what causes those stripes and striations. We have a couple of different features or a couple of different structures within it. The very first one is an I band. If we're looking at one sarcomere, just FYI, is from one little zigzag line here to another zigzag line. So this is going to be one whole sarcomere. So we're just looking at this area. The area where we only have the thin filament. Now, what is the thin filament? The thin filament is actin. So where we only have actin is going to be this kind of purple, bluish color filament there. Where there's only actin, this is the I band. So this area where there's only actin is the I band. This area where there's only actin is the I band. A band is going to be where you have the thick and thin, kind of the area where you have both thick and thin kind of overlapping. So from like here to here would be our A band. We can also see it here, just more of a digital image of those two. And then we have what we call the H zone. The H zone is where we only have the thick filament. So thin filament only was the I band. The area where we only have thick filament, so from here to here, you notice there's only myosin right in this center. That's going to be the H zone. Okay. The next structure is the Z line. The Z line is, I think, probably the easiest of them all to recognize and to identify. Is this kind of zigzag pattern of alternating actin filaments going here. And from one Z line to the next tells you where one sarcomere ends and the other one begins. So from here to here is one sarcomere. From here to here is another sarcomere, and so forth. And the last thing is the M line. The M line, if you were to look very closely at the H zone where these myosin filaments are, there's kind of a little dot or knot right in the middle. That's going to be called the M line. Again, this visual image kind of does a really good job of showing that. Again, this is just kind of going over the same exact things. The only thing that this is, the way that this is different from this slide, so this just has it in word form. Okay. Again, I-band is going to only be where we have those thin filaments only. A-band, the myosin and actin kind of overlapping. So from here to here will be our A-band. H-zone, only thick filaments. So only where we have the thick is going to be our H-zone. Z-lines, those zigzag patterns of the actin. And M-line is the very center. It kind of anchors those thin filaments. Okay. So again, let's look a little bit closer at actin and myosin themselves. Again, these are the filaments that help with muscle contraction. A thick filament is going to be our myosin protein. And our myosin protein has what we call myosin heads. That's these structures right here. These myosin heads want to attach to actin. That's here. And just FYI, again, the thin film is going to be our actin protein, but actin is only these little kind of white ball circles here. That's going to be actin. But if you notice, there's something else that's kind of with actin. Actin isn't by itself. It has other things that are kind of associated with it. So one of the things that we find associated with it is troponin. These are those kind of blue structures right here, the light blue structures. That's troponin. This is troponin, that's troponin. That's associated with it. And we also have tropomyosin. Tropomyosin is this kind of band that goes around actin. That's tropomyosin. Okay? And the thing is, the myosin heads want to bind to actin so that muscle contraction can happen. However, because tropomyosin is in the way, the myosin heads cannot bind to it. So it kind of stops it from contracting because tropomyosin is in the way. So we have to find a way to get these myosin heads out of the way. Okay, so when we're talking about actually contracting a muscle, again, when we think about contracting a muscle, the first thing that you may think about, we have to have some type of connection between a chemical and a cellular level. And when this happens, we are going to have those sarcomeres that are going to, well, the actin and myosin are going to bind to each other. The myosin will then pull on the actin, and they will kind of shorten, basically. I'm going to go back to this slide here. So when these two do join, when contraction does happen, the myosin, which is here, those myosin heads will attach to the actin, and those Z-lines that are here will actually pull towards each other. So they'll move kind of closer to each other, which means that the muscle cell or that sarcomere is going to shorten. And that is why, for instance, if a person decides to flex their bicep, the muscle gets shorter. It doesn't get longer. So when a person is trying to flex or trying to contract a muscle, the muscle gets shorter because the sarcomeres are getting shorter. So that's basically what this slide here is talking about. So the first thing I want to kind of talk or want y'all to think about before we go into actually muscle contraction is what happens when you decide to contract a muscle. What causes a muscle to contract? For instance, if I said I wanted to flex my arm, what has to happen? And I mean, if y'all think, well, just pull up your arm and it will contract. Does it do it on its own? No. You have to think, okay, I want to flex my bicep. And once you think it, then there, of course, must be a signal that has to be sent from your brain to that muscle or the muscles of your arm so that they can cooperate and do what needs to be done for that muscle to be contracted or to be flexed. So that means that there has to be some type of connection between a neuron, which is a nerve cell, and the muscle cell. So there is a very, very delicate and interesting combination or interaction between the nervous system and the muscular system. Everything kind of interacts. Every system interacts in some way. And this is just one way that the nervous system and the muscular system work together. And so the area where a muscle cell and a nerve cell interact is called a neuromuscular junction. We call that a synapse. And a synapse is just a junction between a nerve cell and something else. And synapse is going to be a term that you'll hear quite often, but it's again the area where a nerve cell meets something else, and it could be a nerve cell meeting another nerve, a nerve cell meeting a gland, a nerve cell meeting a muscle. And so in this case, again, it's just a type of synapse. In this case, the motor neuron or that nerve cell is meeting the muscle fiber. And one thing I want to kind of point out, it says where they interact, but it doesn't mean specifically where they touch. Because if you notice here, and even here as we get a little bit closer, those two things aren't physically touching. They're very, very, very close, but they are not physically touching each other. Okay, so the couple parts of the neuromuscular junction, the very first one is the motor neuron. The motor neuron is going to be what brings the signal from the brain to the actual muscle cell. So this is our motor neuron. So it takes things away from the central nervous system to a muscle, to a gland, to whatever it needs to. Okay? And then we have the motor end plate. The motor end plate is just a folded end of the muscle cell, or the folded cell membrane of the muscle cell. So this is the cell membrane here that's kind of folded up on itself and called a motor end plate. And this again is of the muscle cell. So this is where the two things kind of interact. Now I did say again, we have the motor neuron and the motor end plate of the muscle cell. They're interacting, but they're not physically touching. So because they're not physically touching, there is a space between the two. The space between the two is called the synaptic cleft. So again, the space between the motor neuron and the motor end plate is called the synaptic cleft. And as the brain again thinks, this is what's happening, or this is what we want to happen, this is the muscle that we want to contract, it sends a signal via motor neuron, and that signal or that message is carried by structures called synaptic vesicles. You all may remember from taking Biology 1 or whenever you've taken Biology 1 a long time ago or any other science class, a synaptic vesicle is just an organelle that is responsible for carrying something. It could be carrying proteins, it could be carrying anything that needs to be maybe taken out of the cell via exocytosis. So these synaptic vesicles are taking a message to the end of the motor neuron so it can then be released into the synaptic cleft. And the structures or the proteins that they are carrying are neurotransmitters. These are like the message. This is what's going to tell the muscle cell what to do. This is the actual message. So, what is the message? What is the neurotransmitter? The neurotransmitter that is going to be released from the motor neuron is acetylcholine. Sometimes you see ACH for short, but that is the neurotransmitter that's being released. And it's released into this synaptic cleft. Okay? Again, these synaptic vesicles are carrying it. The exocytosis is being released. And so it's in this area. Remember, they're not touching, they just kind of interact. But the muscle cell has to know what to do with it. So there has to be some type of interaction between the neurotransmitters and the muscle cell. Therefore, there are receptors on the motor end plate of the muscle cell that are going to receive the neurotransmitters, receive the message. Once it receives that message, it's going to have a series of steps, definitely important to make sure that you know them and in order. So the first thing that happens after it has received this neurotransmitter, acetylcholine, it binds to the receptor. The very first thing or the next thing that's going to happen is that the permeability to sodium and potassium is going to increase. So permeability, if y'all remember, it means that it allows things to go in and out. And so when that happens, this is going to generate an action potential. Now, usually when people talk about action potentials, people mention this in a nerve cell. But this is actually an action potential that's happening within the muscle cell, which is kind of very unique. So, again, acetylcholine, which is a neurotransmitter carried by synaptic vesicles, released into the synaptic cleft. They're going to then attach to the receptor on the motor end plate. That receptor will then, at that point, that's going to then cause a change in the permeability for sodium and potassium, which will then cause an action potential to happen within the muscle cell. The very next thing that's going to happen is that the SR, the sarcoplasmic reticulum, remember, is responsible for storing calcium. It's going to then release calcium from the SR. This is probably, I hate to say this, but it's probably one of the most important things that's going to happen. If it does not happen, muscle contraction won't happen at all. Of course, if this doesn't happen either, but even if all of this happened, but if there is no calcium release, there would be no muscle contraction. So, starting from the top, so we have to have, again, that muscle fiber and that nerve cell that are going to interact with each other. And I said that if calcium was not released, then we would not have muscle contraction. So if calcium stayed in the SR, that means that the muscles would not contract. And when I say muscle contracts, remember we have this actin strand, and we have this thick myosin strand. And the myosin strand has these myosin heads on them that want to attach to actin. Okay? But remember, going back a few slides, going back to this slide, we have tropomyosin that's in the way. It's actually covering the actin binding sites. And so that means that myosin cannot bind to it. So we have to have a way to kind of get them out of the way so that myosin and actin can bind. So listen up if you're not already listening. What's going to happen next is when calcium is released, it binds to, I'm going to actually come back to this part. So when calcium is released from the SR, calcium will bind to troponin, and troponin will change shape. Again, I'm going to go back and forth to these slides so y'all can kind of see what I'm talking about. So, calcium will bind to troponin. Troponin will then change shape. Okay? Once troponin changes shape, that's going to alter the position of tropomyosin. It's actually going to pull tropomyosin out of the way. So that the binding sites of actin are exposed. So it's going to pull it so that we can now see the binding sites on actin. And now that the binding sites of actin are exposed, now what can happen? Myosin can now bind to actin and form those cross bridges, which is just the connection between actin and myosin. This is an awesome picture that kind of shows everything that's happening here. We have myosin heads that when combined to these active molecules, we have tropomyosin that's in the way. When calcium is released, calcium then binds to troponin. Troponin will then change shape. It will pull tropomyosin out of the way. And so now we see those kind of dark purple spots there. Now myosin can bind to it, which is what's happening here. In this slide, we're seeing myosin actually pulling on actin, which means that, if y'all remember, those Z lines are now moving closer to each other, which means that the sarcomere is shortened, the muscle is shortening. That's what's happening here. We're going to talk about the other things because at this point it's being released. We're going to talk about all that in a second, but that's what's happening so far. Okay, so that's the part of the muscle being contracted. During relaxation, the calcium ions are going to be stored in the XR and the SR, which means that calcium cannot bind to troponin. And so this whole complex is going to be covering the actin binding sites. And so myosin cannot bind to it, so the muscle stays relaxed. I've mentioned this sliding filament model several times already, although I just have not given it a name. But again, when a muscle contracts, these Z lines here are moving closer to each other. The sarcomere is shortening, the muscle is shortening, which means that several structures within the sarcomere are going to get smaller or more narrow. Remember, where we only have actin, that's going to be the I band. Where we only have myosin is the H zone. And so because they're moving closer to each other, we notice this I band and this H zone is a lot more narrow. The Z lines, of course, move closer to each other, and the myosin and actin start to overlap. It's not that the myosin or the actin themselves get shorter, it's just that they kind of overlap with each other. They're still the same length, they just begin to overlap. So, this is going to kind of get us into what happens after it contracts, because we don't want our muscles to just stay in this whole contracted state. We have to have a whole cycle of what's happening from beginning to end, right? And so, if you notice, again, we start here. Actin and myosin are not bound to each other. Actin is covered by tropomyosin, so those actin binding sites are not exposed. Another thing to kind of take a look at is what's associated with myosin. So we talked about how troponin and tropomyosin are associated with actin, but if you notice here, myosin has ADP and a phosphate that's attached to it. And what I like to kind of think about myosin is that myosin always has to have something attached to it. It wants to be with actin, but it always has to have something attached to it. Okay? And if it has actin, then it doesn't need anything else. But right now, it doesn't have actin. So let's go from the beginning. We have calcium that's being released from the SR. It attaches to troponin. Troponin changes shape and pulls tropomyosin out of the way. So now we have those actin binding sites that are exposed, which means myosin can now bind to it. So, since now myosin can bind to it, right here we see myosin being binding to actin. And since it's bound to actin, the next step for myosin is to let go of ADP and phosphate. So remember I said it has to be bound to something. It really wants to be bound to actin. So once it's bound to actin, it lets go of ADP and phosphate. And this is contraction, right? But we don't want our muscles to stay contracted. So we have to have something that is going to allow it to break the bond. Okay? And so the energy that it takes, or the energy that we have to break the bond between the two filaments, is going to be ATP. So ATP comes in. It gives enough energy to break the bond between actin and myosin, which means, again, myosin is attached to something, but it's not actin. But ATP gives it the energy to break that bond. ATP then breaks down to ADP and phosphate, and the cycle continues over and over and over as long as calcium is available. But sometimes we don't want it to just constantly contract, so we have to have other things kind of in place to kind of slow this whole contraction down. So we have to have things that's going to stop this whole process, right? So one of the things that we have that's going to stop this whole process, or to stop the nerve from constantly sending this signal, remember, the nerve sends a signal via synaptic vesicles. Those synaptic vesicles are releasing the neurotransmitter acetylcholine, and acetylcholine is what's going to start this whole thing off. So this neurotransmitter is still within the synaptic cleft. Then we need to get rid of it if we want the neural stimulation to stop. And so in order to break down acetylcholine, we have an enzyme, acetylcholinesterase, that's responsible for breaking down acetylcholine. Now again, why would we want acetylcholine to be broken down? Because we don't want the muscle to constantly contract. So once that's being broken down, all those other steps that we talked about won't happen, which means the permeability to sodium and potassium won't happen. We won't have that neural impulse or action potential. The calcium is not going to be released from the SR, so it's going to go back into the SR and be stored. And since we no longer have any calcium, it cannot bind to troponin, which cannot move tropomyosin out of the way. And so now the actin binding sites are covered, and myosin cannot bind to it, which means that the muscle is now relaxed. So again, if y'all remember, let's go back to this slide here. In order for all of this to happen, we have calcium. Calcium binds to it, so we can have those actin binding sites. ADP and phosphate is released. ATP is what breaks the bond so that this can happen again, right? So we have to have some energy. We have to have ATP. The muscle cells has tons of things in it so that we can produce a lot of ATP. So we have to have several sources of energy, several sources of ATP for this to happen. First, we have tons of ATP that's just in small reserves. It's just kind of hanging out in the muscle cell already. The second way that we get ATP is through creatine phosphate. Now, creatine phosphate is one of those structures that has a phosphate group that's kind of attached to it. And if you all remember, ADP, which is adenosine diphosphate, is just slightly different from ATP in that it needs one more phosphate group. And so that phosphate group that creatine phosphate has can be stripped and turned into ATP. However, the majority of our ATP is through cellular respiration, or is taken in through, or not taken in, produced through cellular respiration. And with cellular respiration, if y'all remember, we have anaerobic and aerobic phases. Anaerobic respiration when it's very low in oxygen, which means we're going to have very little ATP. But aerobic respiration is where we're going to have most of it. We also have myoglobin, which is another complex protein that's found. It has this kind of red structure, red tint to it. We'll talk about it later. That stores oxygen just like hemoglobin stores oxygen, but this is going to be specifically within a muscle. And it's going to be where most of our ATP is going to be produced, or how most of our ATP is produced. Okay? Now, if we don't have enough oxygen, of course, we go through anaerobic respiration. In humans, specifically within our muscle cells, when we go through anaerobic respiration, specifically we'll start to produce lactic acid when we don't have the right amount or the necessary oxygen that's there. Some people hypothesize that this is what causes muscle soreness or even muscle fatigue. And so when we produce the lactic acid, that lactic acid could possibly be converted back to glucose, because remember, glucose is one of those things that we need to start our respiration. So we can get some more glucose, then we could possibly make more ATP later on down the road. But we also need some oxygen in order for that to happen. So the amount of oxygen that we need to take that lactic acid that was produced here and convert it back to glucose, that is called oxygen debt. So again, the amount of oxygen that is needed to convert lactic acid back to glucose so that we can eventually make the ATP that's needed, that is called oxygen debt. Okay, and this diagram here just kind of shows exactly what I was talking about, how lactic acid could be converted back to glucose, stores glycogen if necessary, but with it can then continue to synthesize more ATP. And this also, I was referring to one of the reasons why muscles become fatigued. It could be because of a buildup of lactic acid. This is one of those things that they're just kind of still studying. One that I always find quite interesting, muscle fatigue can happen from just having a loss of desire to continue to exercise. Your muscles can become tired just from that. I think that's probably most of my issue. Also, ion imbalances. Remember, we have to have calcium, potassium, sodium, all those different ions so that the muscle can contract. And so if those electrolytes are a little off balance, that could pose issues. Or even if you're not moving around enough and the muscle isn't able to get the blood supply that it needs, so that it could get the specific amounts of sodium, potassium, calcium, all that good stuff. And then we also have muscle cramps. A muscle cramp is going to be what you all probably have all experienced at some point in your life, just a contraction that you cannot control. And it's this very intense muscle contraction. A lot of people think that it's a result of the electrolytes being unbalanced. And when electrolytes are unbalanced, that could cause muscles to be severely cramped. This could be solved by possibly eating or drinking certain foods that contain electrolytes, like Powerades, Gatorades, bananas, things like that. And this is the end of part 1 for the muscular system.